Kidney disease affects over 850 million people worldwide. In the US, 1 out of every 7 Americans have chronic kidney disease, and up to 70%, yes, up to 70% are not aware of their diagnosis. One of the people is Utah Jazz General Manager, Mr. Justin Yannick, who went in for a routine physical after 8 years and was found to have advanced kidney disease on blood tests. He underwent a kidney transplant about 3 weeks ago and is now recovering well. Besides genetic causes, as in Mr. Zanick's case, the main culprits for chronic kidney disease are diabetes mellitus and hypertension. Treatment is generally directed towards medications and lifestyle modifications such as a healthy diet, exercise, and weight management. Despite these treatments, though, in some patients, kidney disease progresses to kidney failure, requiring either lifelong dependence on dialysis or a kidney transplant. Kidney disease is also a major risk factor for heart disease. Thus, kidney disease is a silent killer, contributing to over 3 million deaths worldwide. Good evening. My name is Nirupama Ramkumar, and I'm an associate professor in the Department of Internal Medicine. As a nephrologist, I take care of patients with kidney disease, and as a scientist, I'm interested in understanding the mechanisms that contribute to high blood pressure and progression of kidney disease. I'm going to take a step back and tell you how I got here. I was born and raised in a small town in southern India called Coimbatore. One of my earliest memories is visiting my grandmother's clinic after she was done seeing her patients. I was fascinated by all the equipment in the clinic, but one thing stood out. It was the blood pressure machine. It had this little mercury that would bob and shine inside the glass case. On my 12th birthday, my parents gifted me this book, and after reading it, I decided I wanted to be a detective. I soon realized that India in the 1990s was very different from Victorian England. And so instead of being a detective, I followed in the footsteps of my parents and my grandmother to become a physician. After medical school, I was interested in pursuing research. I moved to the US to obtain a master's in public health at the University of Utah. It was here that I developed an interest in nephrology, working with my mentors, Dr. Srini Bedu and Dr. Alfred Chung, who are both here. I learned about identifying and modeling risk factors for chronic kidney disease progression, as well as conducting large clinical trials in dialysis patients. Utah also has historical significance when it comes to kidney disease. Dr. William Kolff, a pioneer in the field of dialysis, and who invented the first dialysis machine that was used to treat kidney failure during World War II, was faculty here for 2 decades. Our dialysis unit in Research Park is named after him. Following my master's, I completed my residency in internal medicine in California, but I could not keep away from the beautiful Wasatch Mountains. I returned to Utah to pursue my fellowship training in nephrology. But this time, I wanted to understand how kidneys worked at a cellular level. I worked with Dr. Don Cohan, understanding the genetics, understanding kidney physiology, genetics, molecular biology, and development of genetically altered mice. I love doing basic science research, so after my fellowship, I decided to continue that as my long-term career. One of the systems we've been studying is the renin-angiotensin system. In this system, it's an important regulation of blood pressure as well as salt and water balance. I know this is complicated. It has multiple moving parts. What we've done is to systematically study each of these components in regulation of blood pressure and salt and water balance. We did this by using genetically targeted mice. The advantage to using genetically altered mice is that one, despite their small size, their blood pressure is very similar to human blood pressure. And two, we were able to either selectively knock out or overexpress genes in specific cells within the kidney. Over the last several years, I've been interested in studying a protein called the prorenin receptor. It's a component of the renin-angiotensin system. A receptor, to those who don't know it, is a large protein that's embedded in the cell membrane that receives signals from hormones, chemicals, or enzymes that are outside of the cell. The prorenin receptor is present in multiple organs and receives signals from both prorenin and renin. As you can see here, it's present in the kidney, the brain, fat cells, placenta, blood vessels, and in the heart. Using gene targeting, we developed a novel mouse that lacks the prorenin receptor specifically within the kidney. We did this to study the effects on blood pressure. To study blood pressure, we use a specialized device that is implanted in the blood vessel of a mouse to provide continuous real-time measurement of blood pressure. Shown here is a graph with the average 24-hour systolic blood pressure on the y-axis and time on the x-axis. Normal blood pressure ranges between 100 and 120 millimeters of mercury in mice as well as in humans. Control mice at basal conditions have normal blood pressure in the normal range. However, when they are infused with an external stimuli such as angiotensin II, they develop hypertension that continues throughout the duration of the study. In contrast, prorenin receptor knockout mice that lack the prorenin receptor within the kidney are protected against developing hypertension throughout the study. We went on to show that these blood pressure protective effects in the knockout mice were through changes in both sodium transport and water balance. Our new recent project is to understand the role of prorenin receptor in kidney disease. Since symptoms of kidney disease do not develop until fairly advanced stages, we rely mostly on blood and urine tests. One of those blood tests is the blood urea nitrogen, which in normal conditions is usually less than 25 milligrams per deciliter. Elevated levels usually signifies worsening kidney disease. Compared to mice that have a normal intact prorenin receptor, mice that lack the prorenin receptor had lower blood urea nitrogen levels, suggesting protection from kidney disease. We also use kidney histology to examine at the structure as well as scar tissue, in this case that is shown in blue. As seen here, control mice have a lot more areas that stain blue, suggestive of worsening kidney damage, compared to the control mice that don't have any of this. You can also see dilated kidney tubules, which are shown here in large white spaces, are a lot more in the control mice compared to the mice that lack the prorenin receptor. We're now evaluating the cellular and molecular mechanisms that contribute to the prorenin receptor mediated effects on kidney disease. I recently received an R01 grant from the National Institute of Health to study this. Taken together, these findings show that the prorenin receptor is an important regulator of blood pressure and kidney function, and perhaps could be a target for treating these conditions. Our ultimate goal is to find a new therapeutic strategy that we can use to slow progression of chronic kidney disease and treat hypertension. With that, I'd like to thank my mentors, Dr. Bedu and Dr. Chung, who are both in the audience, for instilling the love for kidneys and for nephrology. to Dr. Don Cohan, who trained me to be a detective in the lab, multiple collaborators that have helped me identify and deduce different aspects of the mystery of the kidney. I see Katsu here, and I know Scott Summers is up there as well, and multiple students, as well as our laboratory team, mainly Deborah Stewart and Will Wheatley, that I've worked with for several years to do our studies. Lastly, I'd like to thank my parents, who are no longer here, but who encouraged me to dream big, work hard, and think outside the box. I'm now proud to be a scientific detective, just like Sherlock Holmes, on the heels of a silent killer.